It's a really, really good question. So there are different types of problems with alignment, and depending on where that occurs, you could have a bigger piece missing or a slightly smaller piece missing. So when we think about chromosomes, a megabase is a million base pairs, so a million of those letters, A, C, G, or T. And when we look at the chromosome 15, where these replicated regions are, those are all the regions where you could have misalignment. So the typical type 1 deletion, which is about 6 megabases, goes from breakpoint 1 all the way to breakpoint 3. So across the whole chromosome 15 in this region. And type 2 goes from breakpoint 2 to breakpoint 3. So it's slightly smaller. It's about 4.5 or 5 megabases for the type 2. So there's not great data about exactly what the difference is between the two. That's something that there's only 2 of those types, yeah. Well, there's one more type that's a little bit, so see how this says rare? You could have an even bigger deletion that's quite rare. But the ones that people are mostly talking about are the type 1 and the type 2. Because that's relatively common, type 1 being slightly more common than type 2. And from our experience, in terms of development, there may be slight differences between type 1 and type 2, with type 1 being slightly more severe. But when we've gotten case series and things together and looked at statistical significance, it doesn't look like it's that statistically significant. Yeah, it's a great question. It's something that needs to be pulled out of some of the data that people have looked at. So when we look at the natural history study, we're just grouping deletions in one big category, but we probably should pull out the data and look at it more closely. Yes? Yes, great. Mm-hmm.
So, but just from hearsay, like some people have said, or parents have said, like it could be like with how big the deletion is,
Mm-hmm.
it could be the severity of everything with the symptoms and whatnot with the Angelman. So, is there any way we like or I think some people have said that they know how big the deletion is, how small, but
Mm-hmm. 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 Okay. Yeah, so on chromosomal testing, it depends how you were diagnosed. So we used to do a test called FISH, which is, yeah. Yeah. So FISH, let me go back to this slide. So FISH is basically a single one of these many probes. So it's getting at almost a single letter. It's a little bit bigger region than that, but think of it as just targeting a single letter and looking for a missing portion. And so early on, we just sent a light to one place on the chromosome, and then we looked at the cells, and we looked to see how many of those lights there were. And if we were missing a light, then we knew that there was a missing part. And now we do chromosomal testing where we look across all of our pairs of chromosomes, so all 23 pairs of chromosomes, and we look to see if there's small missing or extra pieces across all of our chromosomes. And because of these little tiny probes that we use to look at single letters, we can get more information about size that's very detailed. So it depends what type of testing he had, but once you get those results back, the best way to know is just the size of the missing part. So the reports for the chromosomal testing nowadays says you're missing this probe to this probe, which have, it's millions of letters, so it has a long number to another long number, and then it will say the size of it, 4 megabases, which would be type 2, or 6 megabases would be type 1. So it depends on what type of testing you had. You might have that in your file that your child had a chromosomal test that gives you size. Yeah. Just ask them. I mean, because even the clinical trials will want to know that detailed of information. So it's good for all of you to have copies of testing reports, because all the clinical trials will want them. And of course, we don't know what sites will have the clinical trials, but if you have a copy, it's obviously a lot easier to get it to the right people than if we're hunting around for it, for sure. The other thing I want to mention, you brought up Angelman versus Prader-Willi. So you all might know this, but Prader-Willi syndrome is caused by a missing part of dad's copy of chromosome 15. And so all the chromosomal testing can tell us is that there's a missing part, but it can't distinguish whether there's a missing part between mom's chromosome or dad's. It could be either at that point. And so that's where the methylation testing comes in. So once we know that there's a missing part, we still have a next step to look to see and the way that we do that is to look at how many of those, this is simplifying a little bit, but how many of those carbon groups are along, or the methylated groups are along the chromosome of the one that's present. And so the only way to know if it's mom's copy or dad's copy present is to do that extra test. And that's where Angelman syndrome is confusing and unique, because most tests, like if you have a missing part of chromosome 22, you have DeGeorge syndrome, you do the one test, you find the missing part, it doesn't matter if it's mom or dad's missing, you have that particular genetic diagnosis. But because chromosome 15 is imprinted, and you only want to express UBE3A from mom's copy, that's why it's so critical to know which copy it is. And similarly, there are chromosome 15 in this region, you have to have certain genes that are only expressed from dad's copy, or you have Prader-Willi, which has a completely different clinical picture than Angelman. Did you have a question? Okay. Yeah.
How long is the period? If you're talking about several million base pair deletion, side deletion, like how long is the total?
That's a good question. I don't know if I know off the top of my head. It's not megabases though. It's kilobases in length. So it's thousands of bases, not millions of bases. And the reason I know that that's important is because when we package something to deliver it back, if it's a really big gene, it can't be packaged into a virus. So one of the reasons UBE3A is so amenable to gene therapy is the size. But I can look it up for you. But off the top of my head, I don't know that.
That's okay. When you're talking about the chromosomal deletion, we're saying like 5 to 6 megabases. So some of those base pairs are non-coding, or how does that work?
That's a really good question. So this is a really complicated region. There are some small nucleolar non-coding areas. So basically, the gene SNRPN, which is the imprinting control region, is a really, really long transcript. So it's a really, really long gene when it gets made into protein. And if you look across that region, within that gene, in the non-coding parts of that gene, are many different elements that are non-coding. So all of that is encompassed within that space, yes. So when I say 6 megabases for a type 1 deletion, it includes all that non-coding stuff too. Because we're talking at the chromosomal level.
Yeah, I might have some more follow-ups. I might just have
Yeah. Yeah, no, they're great questions. So the confusing thing about genes is that when we make an active product of a gene, so when we take UBE3A, which is just at its simplest form, just those letters, A, C, G, and T, which make up our DNA, and we look at it as part of a chromosome, it has coding parts and non-coding parts, introns and exons. So the introns are the non-coding parts, the exons are the coding parts. But when we get to the point where we're making those proteins, the gene has been what we call modified, and anything that was a non-coding piece has been removed. So that's where we get into the details of coding versus non-coding. No, so the SNRPN gene goes all the way across the region because it's the control region, but if you look at the chromosomal region, there are many different genes within this region, and one of those genes is UBE3A. So it's a number of different genes. So when we're talking about Prader-Willi, there's a number of different genes in that region. For example, there's a gene called MAGEL2, and there's a gene called NDN, and they're all in that region that we're looking at of the chromosome that we need dad's expression from. And similarly, UBE3A is just expressed from mom's copy, and the way that the body controls that is that SNRPN gene, so that's why it has a protein part of it that goes across the whole chromosome there, because it's controlling what's turned on and off. But within that region, there's multiple different genes, and UBE3A is one of them.
So this is the upshot, my question.
Yeah.
Is the variability in phenotypic expression that we see in our animals, which is what she was asking, is it possibly due to the idea that maybe there's like deletions in nearby genes, like nearby genes have been affected as well? And so,
Yeah. Yeah, great question. So beyond UBE3A, there are some non-imprinted genes in this region. So meaning that those are genes that express from mom and dad's copy. And those are actually, so GABA is the inhibitory neurotransmitter in the brain, GABA aminobutyric acid. And I don't know if the pointer works, it doesn't. I think it was working, okay. So if you look here, so these are the genes that are turned on from mom's copy, and that includes UBE3A. But then right here, you see these few genes, and the reason we include them is because they're very important for response to GABA, the inhibitory neurotransmitter in the brain. So we think one of the reasons that individuals who have deletion have more likelihood of seizures is because those genes are missing. Nobody's ever proven that. There are kids who have mutations, so misspellings just in the UBE3A gene, and those individuals can also have seizures, but they're much less impactful and significant seizures in some cases, not all, And so we think that those genes also play a role. Yes, they're expressed from mom and dad's copy. Yes. So, and that's also a really, really good question because there are genetic disorders that are caused by having problems with those genes that cause seizures, but you have to have a misspelling in both mom's copy and dad's copy. They're what we call autosomal recessive disorders. So, UBE3A is a hard example because it has to be from mom's copy, but UBE3A, because it's enough to have just your mom's missing copy, we would sort of call it autosomal dominant, but again, more complicated because it has to be mom's copy. But and so autosomal recessive just means both copies have to have a problem for you to have seizures from it. So the truth, but that's a hypothesis that we all believe is the case, that those genes also play a role. And that's also why we use certain medications for Angelman syndrome for seizures work better than others. Because benzodiazepines, so Onfi, clonazepam, target the GABA A receptor. And so they help with response of that receptor to circulating neurotransmitters, so circulating GABA to tone down the brain a little bit. And we think a lot of the problems with Angelman are related to kind of things being turned up a little, so that inhibition not working well, and kids with Angelman just having that active neurotransmitter or that's very excitable. So a lot of the features of Angelman, seizures, decreased need for sleep, hyperactivity, anxiety, we think could be related to problems with GABA. So the Ovid trial, which I think they presented yesterday to you guys too, that drug targets those receptors too. And that's why the drug was brought to Angelman, is because we think there's a problem with these receptors. Yeah.
So EBD where the gene is just not just specific, right?
Yes.
Silenced on both copies, is that right?
So yes, yeah,
So
you don't have an active copy.
they might not have, right, they might not have such issues with seizures because they don't have like an issue with these specific genes, these GABA genes.
We think. We think.
Okay. So the phenotypic, or so the symptoms that we see in our angels, they're mostly coming from this like UBE3A malproduction of ubiquitin migrans, right? They're not coming from the potential influence for the most part of other genes around. Or it's both.
It's both. It's definitely both, but we have enough data to say that, including clinically, right, because we have kids who just have a misspelling in UBE3A who have a similar clinical picture in many ways to individuals who have deletion. So we think returning, so all the strategies for gene therapy, for example, are focused on UBE3A. However, the ones that turn on the paternal copy, so turn back on dad's copy of chromosome 15, may also impact these other genes too. Because all they're doing with the antisense oligo that was talked about is that they're working on the control region where serpin is. We know that there are certain factors that come in and bind right there, so get stuck on the chromosome, that allow other factors to come in to turn off that region. So they're targeting that. And so that's a chromosomal target. So when this gets turned, the control of this gets released, so it gets basically demethylated, then what we know is what we think is going to happen is that it's going to impact the whole region. Versus the gene therapy that returns UBE3A with a virus is going to just impact UBE3A.
So potentially, that would fix some of the symptoms, it would help with some of the symptoms, but potentially in this example with GABA, not the seizures.
Well, UBE3A is responsible for some of the seizure activity for sure. Yeah. So but again, we don't really know. But a lot of people favor turning on dad's copy because we turn back on the whole region as opposed to delivering back UBE3A for that reason. In people who have subtypes of 2 copies of dad, so uniparental disomy, people who have deleted region of the chromosome, and people who have imprinting control issues, as opposed to people who have just a misspelling in UBE3A, so it may end up that the strategy of turning back on dad's copy is not the best strategy for the people who have mutations in UBE3A alone. So we'll just have to see how that plays out. Yeah, of course. Anybody else have any questions? Yeah. Mm-hmm. It plays a role for sure. That's a great question. So I think a lot of people with Angelman syndrome, for example, have developmental delays and haven't developed speech. And so the goal is that we target the key features and in addition treat some of these additional features that we're now recognizing. So the hope is that it would impact everything, because basically what you're doing is restoring the function of something that wasn't there. And so our hope is that we can restore we can fix problems with walking, fix problems with speech, fix the sensory and the hyperactivity and as well. So we're hoping to target everything. We're going to learn about whether that's realistic, but so I do think, though, that there's potential benefit. Yeah. That's awesome. Mm-hmm. Mm-hmm. Yeah, which is great. Right. Mm-hmm. Mm-hmm. Mm-hmm.
may take a lot of it within that time period of reproduction of pain. And once I started using this simple system, of course, we started to realize, hey, I'm talking to me with this thing or something. He's
Mm-hmm.
in here, that
Mm-hmm.
in here has always been there, but it's like the demeanor has changed. Like, he would still do some driving when he gets excited and we think he wants to get your attention. And there's 2 hours in what we get home because I'm trying to freestyle or whatnot, but he's doing it just slightly more. So I don't know if it starts to maybe with more of that pre-teenagers too. I have a really started to get hard. I'm starting to lose my way.
Mm-hmm. Yeah. It's absolutely. Mm-hmm. Mm-hmm. Yeah. So there's a difference between supportive therapy and the gene therapy. So the goal of the gene therapy is to fix the original genetic change that caused the Angelman syndrome. And that's a very different thing than talking about some of the medications and things that we use to help. But your son is at an age where we often do see an uptick in some of the behaviors potentially related to anxiety. It's something we're still trying to understand. So there may, just hearing you and some of the concerns you have, it may be worth talking about some of the supportive therapies too to see if we can help. Some of which are behavioral things too, but yeah. Yeah. Mhm. Yeah, that will be helpful to talk to them. There may be behavioral things. There may be medications they feel like would help anxiety that could help too. It's hard to know without knowing this more specifics. But I think when we're talking about gene therapy, we're talking about fixing the root of the problem. And when we're talking about some of what's available right now, all we have is supportive things to try to treat symptoms, unfortunately. So, but there's lots of exciting things coming. So. Anyone else have questions about genetics? Okay, well, thank you all for coming.